Hey folks, welcome to another edition of Zog Science. Today we're going to be talking about cells, and we're going to be specifically focusing on how do cells process matter and energy. So before we get started, we need to talk about a couple things. First of all, just a little reminder, both energy and matter cannot be created nor destroyed, but they can be changed from one form to another. And because of this, cells are not necessarily making energy. That's kind of a misnomer. What they're really doing is they're just converting energy from one form into another, and that form that they're going to be converting into that's actually usable within themselves is ATP. So how do cells convert matter? Well, they have a few organelles that kind of help with this. Generally, what they're going to do is they're going to use their lysosomes or their peroxisomes. Now, lysosomes are only found in animal cells, whereas peroxisomes can be found in both animal and plant cells. Lysosomes digest macromolecules and they clean up broken down organelles. We kind of looked at this the last time, but they kind of fuse with them in order to make that happen. And they, again, are little vesicles filled with digestive enzymes. Peroxisomes are pretty similar. They detoxify a cell, and they generally are going to be used to break down fatty acids and sugars. You can kind of see what those look like under a scanning electron microscope. So lysosomes, what they do is they fuse with food vacuoles, and then they break down polymers into monomers, and those monomers now become nutrients for the cell to reuse to build new molecules. Lysosome enzymes work best at a pH of 5, where the cytosol of a cell has a pH of 7. Now, it's kind of interesting. The reason why that is because if the enzymes leak into the cytosol, they're not going to digest the cell themselves. You know, if a few get out, they're not going to be able to work because they're going to denature, and then they won't be able to function. So that kind of goes back to when we were talking about why do we have organelles, you know, the double membranes producing kind of a specific area that has different conditions inside of it. There are some lysosomal diseases, so when your lysosomes don't function properly, these are generally fatal. And what happens is that the lysosomes can't pick up molecules, or they can pick them up, but the enzymes aren't functioning properly. And so because of that, lysosomes tend to fill up with the undigested material, and eventually they impede cell and organ function because they're just becoming very large in the cell and not allowing the cell to do what it needs to do. One example of this is Tay-Sachs disease. It's a buildup of undigested fat in brain cells. So the enzymes inside lysosomes that are supposed to break down the fat are not functioning properly. And so you get lots of fat that builds up in the brain cells, and this can lead to mental retardation because, again, your nerves and neurons are not functioning properly, and generally death by the age of 4. Now, there are times when your lysosomes are functioning properly to kill a cell, and that is called apoptosis, also known as programmed cell death. And the lysosomes destroy cells at the proper time. This is really useful in the developmental stages of organisms. For example, when a tadpole tail is reabsorbed, those are actually cells that are pre-programmed to die. Also, your fingers, if you kind of look at 6 weeks, right, your fingers aren't really showing, but by around 15 weeks, they're starting to show. What's happening is that you've got apoptosis occurring in between. The webs are kind of are being reabsorbed, and the nutrients are being absorbed into the body. If your fingers do not unfuse properly, it's called syndactyly. Which can happen, you know, kind of rare, but it does happen. Generally, they just kind of cut between the fingers as long as the bones aren't fused together. Now, that was converting matter. How about converting energy? Well, we've got two main organelles that are used to convert energy, the mitochondria and the chloroplasts. Mitochondria convert stored energy inside glucose into active energy, or ATP, and chloroplasts convert light energy or sunlight into active and stored energy, ATP, and carbohydrates. Let's look at some of the similarities of mitochondria and chloroplasts. They both transform energy. They both generate ATP. They both have their own double membrane. They're kind of semi-autonomous, so they actually move, change shape, and divide on their own. And you might have seen that with the Elodia, that they move around a little bit on their own. And then they have their own internal ribosomes, DNA, and enzymes. So what are some other organisms that we know about that have those things? Well, bacteria. So we have this idea, the endosymbiotic theory, and remember we've talked about the fact that evolution is kind of this driving theme behind all of biology. And so this theory is in regards to how mitochondria and chloroplasts evolved. And the idea is that mitochondria and chloroplasts were originally free living prokaryotes, which were engulfed by proto-eukaryotes, or kind of early eukaryotes. So we would have had like a eukaryotic cell, or an early eukaryotic cell that has its own nuclear envelope, and some the endoplasmic reticulum maybe, and then it absorbs a aerobic prokaryote that is eventually going to evolve into the mitochondria. And there's kind of a split because now you have another cell that absorbs a photosynthetic prokaryote, and now you've got your animal and plant cells. And there are actually some interesting evolutions of other plastids. Chloroplasts are a class of organelle known as plastid, and you get that looks at kind of plant evolution, which is pretty cool. And these are kind of some of the evidence for it. They're not part of the endomembrane system. They're not involved in that at all. They grow and reproduce separately. Apparently, I can't spell. They have their own ribosomes, and their ribosomes are structurally similar to those found in bacteria, and they have their own circular chromosome. So a lot of evidence that points to them having originally been a prokaryotic cell. So let's take a look at the mitochondria. Its function is to perform cellular respiration or generate ATP from the breakdown of sugars, fats, and other fuels. It requires the presence of oxygen, and you're going to be breaking down large molecules into smaller ones to generate energy. That's called catabolism, and it is the generating energy in the presence of oxygen, which is aerobic respiration. So we're using this process of aerobic catabolism respiration. Let's look at the structure. It's got two membranes. The reason why it has two membranes is more surface area for the reactions to occur. It has a smooth kind of outer membrane and then a highly folded inner membrane called the cristae. And it's got a fluid-filled area between the membranes. And in the internal fluid-filled space is where the mitochondrial matrix is, which is kind of like the cytosol. And it's got its DNA, ribosomes, and enzymes in that space as well. And you can kind of see what that looks like in this picture over here. Almost all eukaryotic cells have mitochondria. Some of them only have one large one, and some of them have thousands of individual ones. Obviously, it kind of makes sense. The number of mitochondria is correlated to aerobic metabolic activity. Basically, if a cell is more active, it's going to need more energy. Therefore, it will have more mitochondria. So muscle and nerve cells have a lot of mitochondria because they're doing a lot of things all the time. That's why people talk about, thinking and learning and reading and things like that actually being, being things that you need food for, right? People say have a good breakfast before a test. It's because your nerve cells have a lot of mitochondria. In order to help them be able to function properly, you need to have enough fuel in your body to be able to run them. We also have chloroplasts, and this is kind of the picture on the right is what I was alluding to in terms of the plastid evolution. You originally kind of have the original protoplastid, which evolved into leucoplasts, chloroplasts, and chromoplasts. And the leucoplasts split up into three different types. Again, chloroplasts are plant organelles. They're not found in animals. They are a class of plant structures that are called plastids. Some of the ones that you probably should know are the amyloplasts, which store starch in roots, which we saw in the cell lab that we did. Chromoplasts, which store pigments in fruits and flowers, kind of giving those nice bright colors. And then obviously the chloroplast, it stores chlorophyll, which is what makes it green and absorbs light waves. And then it functions in photosynthesis. So the function of the chloroplast is to facilitate photosynthesis occurring. It actually generates a little bit of ATP as well as synthesizes sugars. It transforms solar energy into chemical energy, and it produces sugars from carbon dioxide and water. Again, it's semi-autonomous. It moves, changes shapes, and divides on its own. Let's look at the structure. It's got, again, two membranes, increases surface area for the reactions to occur. It's got the stroma, which is an internal filled space. And then that's where the DNA, ribosomes, and enzymes are. Inside that, you've got the thylakoids, which are membranous sacs where the ATP is made. You can kind of see what the thylakoids look like over here. That's going to be the dark. And then the grana are the stacks of the thylakoids, so having each of them kind of stacked on top of one another. So just kind of putting it all together, energy and matter being cycled together. It is the processes of photosynthesis and aerobic respiration that are making this happen. So the sun energy drives photosynthesis, which will create some ATP along with glucose and sugar. The glucose and sugar, and oxygen combine together in respiration to form more ATP, which form carbon dioxide and water, and it just keeps on going around and around. All right, folks, that's all I've got for you today. Have a wonderful time, and I'll see you next time.